My name is Bradley Coleman. I'm a postdoc in the Gubbels Laboratory in the biology department at Boston College. The lab studies Toxoplasma gondii, which is a single-celled parasite that lives inside of a host cell, and the host can be humans or it can be other animals as well. And so I study the different processes and proteins that are involved with the hope that maybe we can identify targets that could be used for drugs. I'm from New Jersey, originally. From as early as I can remember, I was always interested in science in some way or another. And I remember just always really loving biology classes and watching nature shows on television that was what I did. When I was watching TV, I was watching shows about animals. I wanted to be an astronaut for a very long time, as I think most kids do. So I pursued that as a biology major, never exactly knowing what that would turn into. I didn't know any scientists, and actually I think I didn't know what it meant to be a scientist until probably I was partway through college. And I started working in a lab and got to see what research was really like. So I did a rotation in a lab that studied asthma and one that studied cancer genetics. And then the last rotation I did was in a lab that studied malaria, which was interesting to me because it's a parasite, but also because it's a neglected disease that kills people, hundreds of thousands of people a year, but doesn't get a lot of attention. That really struck me, and then the parasite that I study now, Toxoplasma gondii, is a related parasite to the parasite that causes malaria. I study proteins. We actually take the parasite itself and we will mess with the version of that particular protein we're interested in the parasite, which is a bunch of stuff we don't want, but also the stuff that we do want. So what happens after that are a bunch of steps to purify out the stuff that we want. And you create pieces of DNA at the bench that have a certain sequence that can then go into the genome of the parasite itself. So that's the parasite's copy of the gene that codes for the protein. You basically switch one with the other. And so now instead of having the normal version of that gene, the parasite has the version you made. Now we go spin it in the centrifuge. So this is going to spin this tube really fast. The DNA we want is in the liquid stuff. So all that stuff that's going to spin down to the bottom, we're going to throw away. We're going to keep the liquid that floats up to the top. What is it about that particular protein at this particular moment in the parasite's life that is so important, and why does the parasite die when you get rid of it? So that's the basic biology question that we're trying to answer. What's called a transgenic version of the parasite. No two people in the lab are working on exactly the same project, but we're working on different processes in the same parasite. So we can always ask each other for ideas or ask each other for advice. Things start to funnel together. One person does a certain experiment and we think, oh, that'd be a cool experiment for me to do with my protein. And so then I'll try it. And then my results might be similar, but not exactly the same as another person's. And then we can talk about why they're different and why they're similar and what that might mean for the parasite as a whole. And so those sorts of interactions really, I think, further the projects of all the people. I think anybody that works on a parasite or anything that causes a disease, the hope is always that your research is going to translate into a cure or a treatment for that disease, because that is making people sick or killing people. And in my case, it's not just for toxoplasmosis, which is the disease that Toxoplasma causes, but potentially for malaria as well, because it's a related parasite. What I love about being a scientist is discovery, and that you never quite know where it's going to take you. And so it's always exciting and it's always a bit unpredictable. And on top of that, you're sort of making a contribution to knowledge of the world, every time you make a discovery at the lab bench, it's something that no one else has known until you just figured it out. And you can't really beat that.